Okay, I see we have a number of participants settling in to our webinar this morning, so we will wait just a minute or so more and allow everybody to get settled in, and then we'll go ahead and get started. We'll give it just about one more minute, and then we'll get started. Okay, it looks like everybody's settled in. We've got a great attendance today, so we'll go ahead and get started. Good afternoon, good morning, and good evening, everybody, depending on where you may be logging in from around the world. I am Rob Burgess, and I head up business development for Sino-Biological. And I'd just like to welcome each and every one of you to the next installment in our Lock and Key Immunodetection webinar series. In this series, we hear about really cutting edge technologies as they apply to immunodetection, which primarily is related to antibody development and discovery. And we have an excellent speaker today from Aureka Biotech, and we're going to hear a lot about AI as it applies to high throughput antibody discovery. So I think you're really going to enjoy this webinar this morning. We have one housekeeping issue, and that is that we ask everyone who's attending to please type into the chat box, hello, and tell us where you're from, because we're always interested in hearing about where people are logging in from all around the world. Also, we ask you to type any questions that you might have for the speaker into the chat box. We will withhold all of the questions until the end, and then I will walk through those questions with the speaker verbally and try to get to as many as I can. So please do ask some great questions. And so with that, it is my pleasure and honor to introduce to everybody our speaker, Dr. Alon Wellner, who is the Chief Innovation Officer and co-founder of Aureka Biotech. Dr. Wellner is a synthetic biologist with extensive experience in both academic and private sector research. He specializes in protein and antibody engineering through constructing directed evolution systems and is currently leveraging artificial intelligence and machine learning technologies to enhance antibody discovery and optimization processes. Dr. Wellner is currently the Chief Innovation Officer, as I said, and the co-founder of Aureka Biotech, and his academic background includes a doctorate in biochemistry from the Weizmann Institute of Science. And so with that, Dr. Wellner, we appreciate you conducting this webinar today. It is an honor to have you. And I will now turn it over to you so you can begin your presentation. Thank you.
Okay, good morning, Rob. Good evening to everybody, and thank you very much for the invitation. Highly appreciate this. So, I represent Aureka Biotechnology. We are a company that started about 3 years ago. I'll give you a little bit of background about our company. You can consider us an academic spin-off from University of California, Irvine. That's where we developed our own technologies. We've licensed them from UCI, and we are co-founded by these three academic members: Weian, Shongjian, myself. We have a very strong scientific advisory board that consists of Peter Teissier, one of the earliest pioneers that combined machine learning and antibody engineering. Some strong academic, some also strong industry experts like Baitong Zhao, who recently sold his company to Genmab for $1.8 billion. He was the co-founder of Profound Bio, and David Chen, co-founder of Proteologics, sold his company to J&J for almost $1 billion. last year. So we're located in Irvine, California. We're about 20 people. Um, and we specialize in integrating high throughput biology and machine learning. So some background here. Everybody appreciates how hard it is to bring biological drugs into the market. The average drugs takes about 12 years from inception all the way to marketing, with a price tag of about $2 billion. And the fallout rate is about 90%. That's not only very frustrating, but it's also very expensive. And that those costs eventually trickle down into the patients, making these biological drugs very expensive and inaccessible to the vast majority of the population. So our mission at Aureka is to accelerate and this process and bring better leads into the clinic with a higher success rate. So kind of like democratizing the process of drug development. And the way we achieve this is by integrating high throughput biology with AI. So all of our experimental systems are uniquely tailored to generate lots of high quality, high throughput data. And we use that data to train machine learning models. And in turn, those machine learning models predict new libraries, and then we can iterate this process very quickly. And I'll tell you all about that. But this iterative process enables us to extensively uh, search and screen the sequence space of these molecules to find best-in-class molecules that not only have unique binding properties, but also unique functional properties. And then we can go after like the high-hanging fruit in antibody engineering, like multiple modalities. We target very complicated and challenging targets like GPCRs, ion channels. We also specialize in conditional activations such as pH sensitivity, and that allows us to target, to have unique drug targeting, like for the tumor microenvironment or brain shuttles. So our platform consists of three main technologies that we call AuraGlow, AuraQuest, and AuraPicasso. AuraGlow is a single cell functional screening system that's based on microfluidics. So that enables us to screen very large libraries of molecules directly for their function. And I'll give you later some examples. AuraQuest is a synthetic biology system where we highly engineered yeast to basically mimic the function of a B cell. So simply by culturing yeast, we can cover vast landscapes of sequence space and engineer very unique properties into antibodies. And AuraPicasso is our generative AI model. So by iterating between these three modalities, we can very uniquely engineer antibodies with unique functions. So a little bit about our AI engine. So what we specialize is generating our own foundational protein language models. It's inspired by AlphaFold, and that enables us to do structural modeling with very unique precision. We can design very unique sequences and large libraries, and we can also incorporate developability predictions into all the antibodies that we generate. AuraGlow is basically a system that enables us to encapsulate biological reactions into picoliter droplets. So imagine you have a library of a million different antibodies that you want to screen directly for their function. That means that you would have to go over more than 10,000 microtiter plates, right? Which even with automation, this becomes very daunting, expensive, and even impossible, some would say. So what we do is encapsulate this entire experiment. We miniaturize that into a single test tube. And this very schematic illustration here shows how a therapeutic library cell secretes an antibody into the droplet. And that antibody, the activity of the antibody is being reported by a reporter cell that's co-encapsulated into the same droplet. So this is very simplistic illustration. We have very sophisticated experimental designs. Sometimes we encapsulate 3 or even 4 different cell types. But we can capture very unique biology in these droplets. And as long as we can engineer the reporter cells to report with some kind of fluorescent signal, we can capture any type of biology. And with this, we already unlocked many different applications. I'll give you an example of one very important application. But first, I'll just give a brief overview of how the system looks, kind of like a bird eye view. So we have these flexible functional reporter cells. It really depends on what kind of function you want to measure. We're really good at building very large libraries where we develop tricks where every cell can incorporate only one type of antibody and express it very strongly. So we engineer these libraries and we encapsulate the reporter cells together with the libraries. We have different tunable assays that really depends on what kind of function you're assaying for. So we can manipulate precisely the composition of the droplet. And then our first detection point is very high throughput. We can screen more than 1 million droplets per few hours, and that detects the function of the cell. And our second detection point is less lower throughput, but it's higher content. So where we could look at like more complicated phenotypes like cytokine secretion, cell killing, and so on. And then eventually the readout from all of these experiments is NGS. And then we use that data to train the AI model. And then we can iterate this process. So one very useful application that we're applying the system for is ADC discovery. So the most important thing for an ADC is to be internalized into the cell very efficiently. And we've developed a system where the antibody, once it's secreted from the cell, it's being conjugated to a pH-sensitive probe. And once the antibody is being internalized into the target cell, it becomes fluorescent. And we actually turned this method into like a quantitative method. We can measure the level of internalization, and this is very effective for bispecific ADCs, also biparatopic ADCs, and also bispecific lysosomal degraders. This is just an example of an established HER2 antibody called zanidatamab that internalizes very readily when it binds to HER2, and you can see that a non-internalizing HER2 antibody does not generate any fluorescence, while zanidatamab generates lots of fluorescence, and we can capture this with our detection system, and this is just an example of how the droplets look like. And as I said, we've turned the system into a quantitative system. We can measure different levels of internalization. So we don't only look at the level of fluorescence, but also we developed algorithms that detect the shape of the peaks that we're detecting, and then we can sort these different levels of internalization into different bins. And this is just an illustration of 3 different bins, but we can actually do this into like many more than 3. So here's an example where our first case study, where we built a library of biparatopic ADCs towards a very common target, a tumor-associated antigen. So we started this with animal immunization. We've immunized common light chain animals. So to build a bispecific, and it's bispecific where each arm binds a different paratope. So it's a biparatopic ADC, and we've isolated 200 different unique antibodies. So we built a library of 40,000 members. So imagine generating 40,000 different antibodies and testing them for their internalization level. That's kind of like a daunting task. Pretty impossible. So instead, we built that library by using molecular biology, integrated it into our landing pad cells, and assayed them with the system for their internalization. So We looked at different levels of internalization, but we also looked at different rates of internalization, so we could both assay for fast internalization or slow and persistent internalization. And we binned and sorted all these cells and NGS them. And this gives us a very detailed map of the internalization level of all of the different members of this 40K library. And this enables our AI to start looking at different parameters of what actually drives successful internalization. So we can look at the geometry between the two different binders. We can look at their affinities. We can start looking at their CDR clusters and different properties like charge clusters and framework class, the distance between the two arms, and so on. And it actually gives us insight, or the AI, into what are the design principles behind the successful internalizing antibody for this target. So this is something that a human cannot actually handle this amount of information, but our AI algorithms can turn this into very useful data and to actually predict sequences that are outside of the training set. So we nominated strong internalizers that some of them are actually outside of the training set that did not exist as part of the collection of 40,000 members that were in this library. And you can see that some of them exceeded the benchmark by over 300%. And just by handpicking, so the partner that we worked with just handpicked some molecules from that library, and you can see their function is much less inferior than what the AI couldn't predict. So I'll shift gears now to our second system called AuraQuest. This is our synthetic biology system that's actually more focused on binding than on function. And I'll give a brief explanation of the design of the system. It's actually pretty simple. This is a yeast surface display system. So this is a yeast cell that displays an antibody on its surface using the canonical AGA1, AGA2 system. So this is very normal. But the unique property of this system is that the antibody is being encoded from this special plasmid that's located in the cytoplasm of the yeast. So this plasmid is a highly engineered synthetic plasmid that encodes the antibody. And it's being replicated by its own orthogonal DNA polymerase. And this polymerase has been engineered to have a mutation rate which is much higher than the mutation rate inside the nucleus. So it ranges between 100,000 to a million-fold higher than the mutation rate inside the nucleus. So basically, once you cloned your antibody or an antibody collection into this plasmid, all you need to do to expand the diversity of this library is to simply grow the cells for a couple of days. So in a 1 milliliter tube of about 10 to the 8th cells, that takes about 2 days to grow, you actually cover all the single and all the double mutants of a typical antibody fragment, like a VHH or an scFv. So again, you clone an antibody, you grow the cells simply by growing to diversify the antibody. And then you can apply FACS selection. So using a fluorescently labeled antigen, you can sort for antibodies that have better binding properties. But I guess the strongest thing about the system is the iteration. So after the cells recover from the FACS selection, they again rediversify the antibodies. So you can iterate this process as many times as needed, because we all know that evolution is a stepwise process. So we need to iterate many times. And in normal yeast surface display, what you would need to do is extract the DNA, rediversify it, retransform it, and that takes a long time. It also is very laborious. In our case, all you need to do is FACS the cells and grow them. So it's also very scalable. And because it's very scalable, you can actually generate lots of data to train AI. And this is kind of what I want to focus on in the next example. So in this example here, we partnered with our collaborators from Ability Therapeutics. Abilita is a company that specializes in generating GPCR antigens. They actually apply directed evolution to the GPCR itself to expand and increase its yield. In this unique example here, they went for CCR8. CCR8 is a notoriously problematic GPCR. It's really hard to overexpress it and to generate immunogens. So by applying their directed evolution to the GPCR, they were able to generate very large quantities of CCR8 in nanodiscs as an immunogen. I'll just mention that when they applied their technology, they increased the yield of the GPCR without affecting its function. It's part of their technology. So CCR8 is a very important GPCR to target because it's overexpressed on Tregs in the tumor microenvironment. So by targeting CCR8 and Tregs, you can target Tregs for depletion, which is a very established mode of action for lots of solid tumors. So by having this increased level of immunogen, Abilita were able to immunize llamas, generate very potent VHHs. The VHHs are actually very unique because they bind the ligand binding cavity of the GPCR. Most antibodies bind the N-terminal loop, which is highly sulfated. It's prone to proteolysis, and it's very variable. So these VHHs are very unique. The problem is that they required humanization, and that really lowered the affinity. So we partnered with them to compensate for that affinity loss that resulted from humanization of this antibody. So this is an example of how AuraQuest works like. I'll just take a small sip. For one of the VHHs, you can see that in the first cycle, we actually apply a high concentration of the antigen, 100 nanomolar, and you can see very weak binding. But nonetheless, we apply this gate here. We sort the cells that are to the right of the gate. On the x-axis is binding, and the y-axis is VHH display. And in the next round, you can see that although we cut the antigen concentration by half, the population shifts to the right. And you can see here actually there's two populations. So this population most probably signifies a new mutation that's taking over the population. And typically at every cycle, you can see that although we decrease the level of the antigen, we get a shift to the right. So after only 12 days, we're able to affinity mature this VHH significantly. But this entire process, it's barely any work. All you have to do is grow the cells and fax them every few days. So because of that, we can do this experiment dozens of different times in parallel. And it's very interesting to note that if you take the same antibody and you affinity mature it many, many different times in parallel, in many cases, it's very non-deterministic. You would not get the same result. And you can actually tweak that to make sure that you're going to get different results. And that by itself generates lots of training data for AI. So we took all the evolutionary trajectories, all these parallel evolutionary trajectories, and we NGS'd using deep sequencing, and we got lots of information. We used that information to train our AI model and to kind of like prompt it to give us sequences that are actually ideal and outside the training set. And that's exactly what it did. It gave us some sequences that are actually unique sequence compositions that are kind of like a synthesis between the different experiments that we did. So this is cell binding data. This is kind of like the final experiment that we did together with them. And I'll kind of walk you through the slide here, this graph. So in gray here, we can see the parental molecule. And then this is the result of humanization. You can see a very steep drop in affinity. And this is our compensation effort. So you can see that after the AI molecules actually outcompete the parental, and that also outcompete the clinical benchmark. So this clinical competitor is a molecule that targets CCR8 that's already now starting phase 3 clinical trials. So as far as we know, these are the best, most potent CCR8 warheads that exist. Our next case study has to do with engineering balanced affinities. It's actually a dual binder to preserve or to combat muscle atrophy. So we all know that GLP-1 agonist causes significant weight loss, and they're very popular, but they also result in unhealthy weight loss. And a very significant portion of the weight loss is actually lean muscle mass. And there is a strong need for therapies to preserve lean muscle mass with GLP-1 therapies. So we identified an antibody, um, which I cannot disclose its identity, but it binds two different targets, and these targets have to do with muscle atrophy. They actually blocking these targets would actually result in increased muscle atrophy and increased lean muscle mass. So biochemical and genetic evidence suggest that by increasing the affinity of this antibody towards target A would increase its potency in vivo. So it binds one of the targets, target B, with an exquisite affinity of single-digit picomolar. And although it binds its other target with a pretty good affinity, if it's sub-nanomolar, it needs to be higher. So our design criteria for this antibody was to increase the affinity also to by at least 10 or 20-fold, while preserving the affinity towards the original target, which is target B. So in this case, we actually knew the structure, the co-structure of this antibody against both of the targets. So we started with an AI first approach. So in this case, the AI looked at the sequences, looked at the structures, and determined a library. And this library is a combinatorial library where we determine different positions in the CDRs. A couple of positions were actually also in framework regions, um, and determined certain vocabulary for each of these positions. And by shuffling all of this vocabulary together, you get a library of about 1 million variants. So we cloned this library onto our yeast display system, and then we further expanded the diversity using the hypermutation. So, and then we took this library and we sorted it into different bins of affinity against both of the targets, and we sequenced all of these bins together. We took all this data and we used an iterative approach to train the AI model to predict a new library, and we iterated on this process a couple of times. And then all this data was able to generate a application-specific model that can tell us what, for a very large cloud of sequences, what is the affinity towards target A, the affinity towards target B, and the affinity towards both, right? And then we could prompt this model to look for antibodies that have a balanced affinity between the two antibodies. And all we needed to do at the end was to generate a handful of molecules. So it was about maybe 15 molecules that we generated as soluble IgGs, and we tested them using SPR. And to our delight, the affinity towards target A increased from 500 picomolar to about 15 picomolar. And surprisingly, we also increased the affinity towards target B. So this is an effort that, would have taken you years, but, using our very high throughput platform, we could achieve this very quickly within 3 months. We went on to test these molecules in vivo. So you can see that in combination with semaglutide, semaglutide on its own results in a very significant decrease in fat mass. With the benchmark molecule, you can see that there's a little bit of additional decrease in fat mass. But with our molecule, you can see a much greater decrease. But this kind of like was the most exciting part for us, where semaglutide on its own results in a significant loss in lean mass, the benchmark molecule prevents that. But our molecule actually generates um, an increase in lean mass. So these mice are not only losing fat, but they're also gaining muscle. And we're now heading with this molecule towards additional proof of concept experiments. This molecule is also highly developable because we incorporate the developability into our algorithm. And kind of the last thing that I want to touch in this webinar today is a very cool application that we're very excited about. So we can essentially take, I wouldn't say any antibody, but we tried this for many types of antibodies and it works very well into a pH-sensitive antibody. So sweeping antibodies are antibodies that can actually recycle through the FcRn molecule system. So if an antibody is able to kind of like dissociate from its antigen, that antigen could be a soluble target, it could be a membrane-bound target, it can actually recycle back through the lysosomal compartment back outside of the cell and capture additional um, targets. And this is very useful, especially for I&I targets, where there's a high burden or a large sink. And there's for antibodies that have a very problematic TMDD, this can extend their half-lives from weeks to months. And so because of our because that the AuraQuest system is so scalable and so quick, so rapid, we can very quickly iterate between different pH conditions. So we can select for binding at physiological pH for potent binders, and we can select against binding at low pH, at lysosomal pH. So we can iterate between this process every few days, generate lots of data, and train AI models specifically to kind of capture what are the properties of pH-sensitive antibodies. Excuse me. What's very unique about the system is that it captures amino acid substitutions that are beyond histidine substitutions. Actually, very non-trivial that you would never imagine that these kind of mutations actually result in pH sensitivity. And we were able to show that we can increase the PK of many different molecules from weeks to months. We've applied this system already to several different antibodies. And what we hope to do in the upcoming year is by applying this to enough antibodies to kind of like build a generalizable model that could predict um, substitutions that can turn any antibody into a pH-sensitive antibody without us needing to even apply the experimental system. We'll just need to validate that. And we're getting close to that. The system that we developed is called AuraSwitch. We take a complex 3D structure or a computationally predicted structure of the antibody and the antigen together. We apply our model that designed the combinatorial library. And then that combinatorial library is further expanded using our AuraQuest system, and then we can iterate between the two different pH conditions, generate this NGS data, and eventually generate these pH-sensitive antibodies. This is just a quick example of how we applied this to TNF alpha binding antibody, adalimumab. Uh, After a couple of iterations, we were able to increase the dissociation rate by 230-fold at pH 6 without affecting the KD at pH 7.4. So kind of to summarize our platform, we are uniquely positioned to generate multi-specific antibodies, agonistic antibodies. These are examples that haven't shown, and also GPCR modulation, pH sensitivity, internalizing antibodies, and co-localization. This was actually one of our first applications for T-cell engagers against CD19. And the way that we work as a company is that Sorry about that. We are actually a therapeutic company. We just closed a round A, and we're developing our own drugs. But since the system is so scalable, we are looking to monetize it, and we are still partnering with pharma companies to generate value for us and for them. And with that, I would like to thank you and hope there's some interesting question and discussion that we can take further. And Rob, should I show this slide now or later?
Yeah, go right ahead if you don't mind, Alon. I appreciate it. Just leave it up there if you would.
Okay.
And thank you for that excellent webinar. We appreciate it. Very exciting technology. Obviously, the application of artificial intelligence for antibody discovery and validation as potential therapeutic candidates. Really, really fascinating work. We have plenty of time left, so I would like to ask the audience, in which we had a very good turnout, if they would please type in their questions into the chat box so that we can get to them. We do have one question so far. It's actually from a Sino employee based in Europe. And I think it's more of a clarification rather than a question. Alexander Jansma had asked about the panel of 40,000 antibodies that you actually screen. And it was a little bit unclear if Aureka developed that panel or if that was brought in externally, and like where did that panel come from that you're actually screening to kind of determine the antibody candidates?
Yeah, so that was a partnership with a biotech company, a large biotech company we worked with. So again, the experiment itself was to they took that tumor-associated antigen, immunized common light chain animals,
Mhm.
and they got 200 unique VHs. So because of the common light chain, we're able to like generate this 40K library of light chain of heavy chains
Mhm.
with the common chain. Yeah, it was a partner.
Great. Very nice. It's nice to have partners like that, can supply libraries like that. So that's good. Sandra Capurro is asking, could you explain more detail about the gain of lean muscle with the new technology? And I'm assuming that's the antibody you identified, while also having weight loss results. And these are in mice, obviously. I was also very fascinated by that. It's very well known that these GLP agonists do cause loss of not only fat, but lean muscle. And so for you to come up with a candidate that increases muscle, but also decreases fat, it's very, very exciting. Could you just elaborate a little more on that candidate?
Yeah, so unfortunately, I cannot disclose the target itself and the benchmark antibody we use for engineering. But this is a kind of like a well-known mechanism for muscle atrophy by blocking certain ligands that actually promote muscle hypertrophy. So it's kind of like a mechanism that combats the decrease in lean muscle mass. The increase in the loss of fat mass or the additional loss of fat mass was actually surprising to us. It wasn't that significant. I guess that the result that we're mostly kind of like excited about is the increase in lean muscle mass. But the biological mechanism is something I can actually not go into because of the confidentiality. Sorry about
I
that.
don't blame you. I wouldn't reveal that trade secret either, honestly, because that's a big one. Yeah, it's interesting, like Wegovy and Ozempic, and I don't think a lot of people know this, but they don't just cause like fat loss, they cause muscle mass loss, and I think it can be pretty devastating to the body. So,
Yeah, especially with people in their 40s, 50s.
mm,
I think sarcopenia is something that we all have to
yeah.
deal with anyway. I think it's a big problem. People are not aware of this, but men at their after their beginning at their 30s lose between 3 to 8% in their muscle mass every decade.
Mhm. Right.
So I mean, you go to the gym when you're 20 in your 20s and you do a couple of reps and you're jacked, right? And in your 50s, it's much harder to gain any. you're just fighting against time. You're fighting against the
Oh, you're
loss of
right.
lean muscle mass, right? So, taking a drug that actually causes sarcopenia, additional loss in lean muscle mass. So, I mean, the short-term gains of Ozempic are clear. I mean, you just lose your
Yeah,
appetite, you're not able to eat. But eventually, I think muscle mass is a very important metabolic organ that for one, am not willing to lose.
right.
Yeah.
Now you're preaching to the converted on that. I go to the gym and I see these high school kids and people in their 20s, and I wish I could retain the muscle mass I'm trying to put on, but I get it. So, uh, very, very good point there. Zach Dobson is asking, I'll just read it word for word. I think it's easier this way. It says, "Hi, Alon, great talk. You mentioned how quickly the droplet assay platform can be implemented. I was curious how much of that speed depends on already having the biology worked out. If you were starting with a completely new target or cell type, is the droplet screening plug and play, or does it require a meaningful like assay optimization phase?"
I mean, this is an excellent question, like spot on. And yeah, it's not plug and play at all. I mean, I think one of the unique things about our company is the fact that our engineering team works hand in hand together with the assay development team to set up a new application for the system. Does take a significant effort. Historically, it takes about 2 to 3 months to set up. Uh, so how do the cells live together in the droplet for an extended amount of time? I mean, for some applications, they need to co-incubate together in the droplet for 1 or 2 hours. For other applications, it could be all the way to 48 hours. Uh, what are the specific conditions that enable them to work together for this antibody secreting cell to secrete enough antibody, for the reporter cell to be healthy enough to report enough function? There are lots of little nuances that have to be solved. So, yeah, historically, I can tell you in the past almost 3 years, we've already unlocked 4 to 5 different applications very successfully. And then uh, with every new target using these applications, we can just plug and play. But for a new application, yes, it does take some R&D.
Great. Thank you, Zach, for that excellent question. John Collins is asking once again, about the 40,000 antibody library. And his question is really simply, how many typically advance to the next stage? And what key steps do you anticipate before a lead actually enters into a clinical trial?
So how many advance to the next stage just really depends on the positivity rate of that library. Specifically with this library, I can tell you it was a needle in a haystack. But also what we were looking for is not something binary. We're looking for like a high level of internalization. So there was probably like 10% of the library did internalize. We were looking for the antibodies that exceeded the benchmark. In some applications, we can see like a large positivity rate. In other applications, it's really looking for the needle in the haystack. So if you have a 40,000 member library and the positivity rate is 0.01%, then you would have to run this library many different times. You would have to cover the library many different times. So you have to like maybe run 4 million different cells to be able to cover the library extensively. Um, how the second part of the question, right, was how long does it take these molecules to reach to?
No, I think it's what are the key steps do you anticipate before a lead enters a clinical trial?
Yeah, so I mean, obviously we will need to make that antibody as a soluble antibody, a recombinant antibody, and test it and validate that in vitro assays. I can tell you in like most of the cases, what we're getting in the droplet can replicate into in vitro assays and obviously in vivo, right? Um, but one thing that we already solved that's built into the system is developability. So these antibodies are highly developable. They have really good biophysical properties. I haven't shown that, but they have really good melting temperatures, hydrophobicity, self-association. All this is built into the system. So it kind of like fast tracks these molecules. We have many different hits, so we can funnel down. Obviously, in vivo is always something that's limiting. We don't have endless amount of mice and monkeys, but our leads typically make it really well through these stages, and that kind of like what makes a successful preclinical candidate.
Mhm. Great. Thank you for that. And Sumaya Ramesh, I think, forgive me if I mispronounced your name. This is a good question. It actually kind of switches gears a little bit and focuses on your technology as it applies to proteins that are not considered antibodies. And, have you considered applying your platforms to improving native proteins themselves in certain signaling pathways, as some of these native proteins could be possible therapeutic candidates? Are you only focused on antibodies, or are other proteins also kind of in your arsenal or in your focus?
Yeah, that's a great question. So, in my previous academic position, I did apply the system to different types of proteins, with all kinds of therapeutic applications, biotechnological applications. At Aureka, we focused on therapeutic antibodies. This is our business model. But I'll be happy to, you can shoot me an email. I'll be happy to further discuss this.
Great.
Yeah.
So we'll just wrap it up with one more question. It's for me, really. I'm just curious. I know you mentioned this a bit, but I'm curious about your business model. I know you have obviously a number of very exciting candidates in early stage research. And you also mentioned that you're partnering with other companies like pharma, biopharma, biotech. Are you considering also a revenue stream based upon like CRO-based services for other companies, where you would implement your platform to help them identify candidates? Or is it more your own internal focus candidates and then more partnership based?
Yeah, I would say that 80% of what we do is our own therapeutic
Mhm.
molecules. That's what drives the most value for the company,
Mhm.
for investors in the long term. But we are interested in monetizing the system to help others, um, but not as a CRO service, more like a partnership model. We want to have some kind of like skin in the game. We want
Right.
to be, have some milestones, royalties, and so on. And the we're very selective about the partnerships that we do. We work with companies that there's very good business alignment with them downstream. But what we're
Mhm.
looking for scientifically are challenges that can, take our system to the next level, like
Mhm.
unlock new applications.
Mhm.
We
Great.
we don't we're not interested in doing what we're already good at doing just for making money.
Right. Right. Sounds great. Sounds like a solid business model to me. We have one more question from the audience. John Collins has another question. This will be the last one. We'll start to wrap things up. John is asking, what human relevant features in the design and validation do you intentionally maintain during the discovery phase to ensure that candidates translate well to the clinic?
Yeah, so I think that's something that everybody is dealing with, which is, the key problem. One of the key problems, I guess, is immunogenicity, right? How do you make an antibody that you can inject into people wouldn't result in an immunogenic response, right? So I think we struggle with this like everybody else. I don't think anybody has like a really unique and good, successful model for predicting immunogenicity. We do our best to calculate the humanist scores of antibodies. The thing is that, there's no high throughput system that can actually measure immunogenicity. There's, our philosophy, and I think everybody agrees with that, is AI is only as good as the data that's used to train it, right? So, um, but we have some projects internally of being able to determine immunogenicity in, I wouldn't say high throughput, but medium throughput, and calculate humanist scores in parallel to that. So that's kind of like the best that we can do at this point.
Mhm. Okay, great. Thank you for that. And I think what we'll do here is we'll go ahead and wrap everything up. Before I forget, I do want to mention our Cell Signaling Breakthroughs Symposium, which will be held on Saturday, April 25th in Seattle. And this will be a symposium of, I don't know, 8 to 12 speakers that are worldwide leaders in cell signaling, and specifically cell signaling cascades and communication, both internally with cells as well as cell-to-cell communication. Again, it will be on Saturday, April 25th in Seattle, and everyone who registered will receive a specific email invite from my colleague Kayla after this conclusion of this webinar. So I ask you all to please consider attending this. I think you'll really find it worth your while. It'll be very valuable for those interested in cell signaling, cell transduction, and cell communication. And so with that, I just want to say thanks to all of the attendees. We had a huge turnout today from all over the world. I just have to mention where everybody's from. And specifically, we had folks from Saudi Arabia, as well as France, Switzerland, Sudan, Germany, South Africa, and Tunisia, and all across the United States. So we really appreciate all the attendees for attending today and the great questions that we got from the attendees. Also, Alon, I want to thank you for a fantastic webinar. Honestly, I think it's one of the best that we've ever had. So we appreciate your time and efforts today. And I want to congratulate you and the team at Aureka Biotech for all of your early success. And I know the company is just going to do extremely well in the future.
Thank you very much, Rob. Appreciate it.
And finally, I just want to thank my colleagues, Kayla Sarmiento and Brooke Beam, for once again flawlessly executing a wonderful lock and key webinar in our series. And so thank you for that, Kayla and Brooke. And with that, I think we will sign off, and I wish everybody a good afternoon or good evening.
Bye bye. Thank you.